So we have how the microscope work, and this slide we already work in this slide last class. Here we have some objects that without a microscope or a magnifying lens, we couldn't be able to see. So we have the peel of an orange. Here we have a feather from a bird, and then we have the wings of a butterfly. Okay, and here we have our magnifying glass. That is how this object is called, magnifying glass. So most microscopes focus light through lenses to produce a magnified image, and other microscopes use beams of electrons. Magnification is one of the two most important things that microscopes do. Magnification is the ability that the microscope have to make us be able to see an image, because that is why we can see a very very small object, and we can see it clearly. So
Can you repeat what magnification is?
magnification is what make us able to see very small objects. They appear bigger. Appear. It's not that they make them bigger. The objects appear bigger through the microscope because because they are magnified. Okay? So the compound microscope, that is the common microscope, the one that we can go and buy because they are not very expensive. They are in the range of price that anyone can buy. They magnify the image using two lenses at the same time. Okay. One lens is fixed in the eyepiece. We're going to see the image of the microscope and we're going to recognize the parts. So one lens is fixed in the eyepiece. The eyepiece is where we put our eye on. And a second lens is chosen from a group of two or three lenses on the revolving nosepiece. That is the part that you can move, so you can choose the lens that you need, because each of those lenses has a different measure. Okay, let's go to the other one. So here we have a microscope, and here there are the eye lenses or ocular lenses. Here's where you put your face. And then you have the objective lenses. These are two or three, and each of these lenses has different measures. So that means that each of them allow you to see bigger or smaller according to what your need is. And then here is the stage. This is called a stage, and this is the slide holder, and between the stage and the slide holder goes the slide. The slide is the glass piece where we put the object that we are going to see through the microscope. And here is the light, and there is a switch because you have to turn it on and turn it off because of the light. And then here most of them have the light intensity control, this part here. Okay, and with this part we can move the revolving nose piece. Okay, we can put it near or far from the object. These are the these are the objective lenses. Here we have them in real life. Okay, and each of them has a different size and a different measure. Here we can see them, and here you have this slide that is made of glass, and on the slide you put the object that you want to see. Okay, so this I already told you. So each of the lenses has a different magnifying power, and by turning the nose piece you can select the lens you want, and then you have the glass slide on the stage that holds the object to be viewed. Okay, so this is magnification. Is the ability to see objects through a microscope and they look larger than they really are. Okay, they look larger, they are not larger, they only look larger. And this is what magnification is called. And then there is another thing that is called resolution, and the resolution is what makes us able to see objects clearly. So that way we can see small details and we don't see them blurry. It's like when you are in the class and you use the in focus and then the image is blurry and you have to move this part that is around the lens so you can fix the image and you can see all the details and you don't see the image blurry anymore. That is resolution. So we have magnification and we have resolution. Okay, here is what resolution is. It is the degree to which two separate structures, let's suppose that we have cells, okay, living cells. Without resolution, we wouldn't be able to see the two cells clearly. We can see, maybe we can think that it's only one, but because of resolution, we can see the two structures clearly and we can tell that they are two different things. Better resolution shows more details. Okay, here we have an example of, this is a poor resolution of a cell, and this one is a cell that has a higher resolution, and therefore we can see is this one with more clarity than this one over here. Okay, so that is the difference between a poor resolution and a proper resolution that allow us to see clearly. And then we have the electron microscope. Electron microscopes, they are big, they are connected to computers, and they allow us to see images that without this kind of microscope we wouldn't be able to see. So the making of these electron microscopes started in 1930, and they use a beam of electrons. You already asked me what beams of electrons are. Son columnas de electrones. Beam of electrons instead of light to produce magnified images. So here we have okay, this is an electron microscope, one electron microscope, and they normally are connected to computers and other things in a lab. So this, for example, is one image taken from the view of an electron microscope. These are the eyes of a fly. So you can see all the details. it has a very high resolution and good magnification too.